Hello there, my name is Matthew Price and welcome to my presentation. Today we're looking at the secretion patterns of salamander biofluorescent skin glands. So looking at biofluorescence is the absorption of higher energy wavelength light followed by the re-emission of lower energy light. So many salamanders have these skin glands which are biofluorescent and they emit this green afterglow. And the function of these biofluorescent glands is currently unknown, but we are looking at these other glands that we can see in the histology called modified Grannon glands, which contain these neutral carbohydrates. We believe that these are analogous and they're implicated in pheromonal signaling. So, to understand the purpose of these biofluorescent glands, we need to know what stimuli causes these modified Grannon glands to secrete. And as a first step, our goal is to determine if transcutaneous electrical stimulation could cause secretion in biofluorescent glands. So we believe after a very light electrical stimulation to the tail, that there will be smaller and fewer biofluorescent glands present. And after a light electrical stimulation to the tail, modified Grannon glands, which are thought to be analogous with its biofluorescent glands, should be emptied. So with these Grannon glands, we believe these secrete antipredator compounds, which will also empty. So looking at the methods, uh, there is a light electrical current applied to the base of the skin of the male and female Desmanathus ocoee salamanders to stimulate these myoepithelial muscle cells around the gland to contract them and induce glandular secretion. The controls were undisturbed. To assess the biofluorescent glands, we use fluorescent microscopy to collect images before and after the electrical stimulation, basically pointing a blue light at the skin to highlight these biofluorescent glands and to see if the biofluorescence is altered before or after. To look at the modified Graham glands, the tail skin was fixed, sectioned, and then we stained it to reveal these skin gland types. The number of these full or empty glands were measured, and the modified granular glands, and mucus glands were shown, and results were analyzed with a program called SPSS. To look at the results, we can see under the biofluorescent glands that each dot indicates a salamander before and after, and they're connected by a line to see the density or biofluorescent gland size increase or decrease. For the controls, they say relatively the same, and also for the stimulated, which are highlighted in blue. The electrical stimulation didn't actually have an effect on the density or size of the biofluorescent glands. The salamander before and after treatment did not have a significant change in size or density. And then we look at the modified Grignard glands. Looking at the histology, we can see multiple empty Grignard glands and the full modified Grignard glands. This indicates that although the Grignard glands emptied, the modified Grignard glands did not secrete whatsoever. The secreted fluids that were present during the experiment were most likely from the Grignard glands. And then we look at discussion. So our results indicate that the transcutaneous stimulation did not appear to have a myepithelial effect on the myepithelial cells surrounding the biofluorescent glands and modified Grignard glands. These glands simply do not appear to secrete actively. Our electrical stimulation was effective at causing the Grignard glands to empty, showing that they were the sources of secretions that emptied in the stimulated treatment group. And the mucus glands also did not really empty through the electrical stimulation. In the process, we also found a subset of Grignard glands that appeared to be caudal courtship glands that are found in other species, so that was notable. To conclude, our results do not support the hypothesis that electrical stimulation causes emptying of biofluorescent glands or modified Grignard glands. The function of the biofluorescent glands and modified Grignard glands remains a mystery, but this is certainly a piece of the puzzle. So electrical stimulation did not cause either of these to secrete. Thank you. I hope you all have a wonderful day. Goodbye.